Here we'll study about chromatography. It seems like a big word, but it's something that you see very often. If you have a piece of paper, and on this piece of paper, if you drop some ink and let it dry, and a little later you add a drop of water onto this paper, you wet this paper with a drop of water, and you keep it for some time, what will you observe? You will observe a picture like this. You will find the ink this black ink has smudged, and now to your surprise there is not just black ink, there are so many colors in it. So what can you say about black ink? Is it just made up of the black dye? No, black ink is a mixture of many colors. In fact, what has happened here is chromatography. These are the two pictures, like this one and this one, is also examples of smudged ink. Ink on which there is a drop of water. Like here, it's very easily we can see that the strips of paper are dipped in water, and as the water rises up, it smudges the ink. All these are examples of chromatography. Let's get into the different steps of chromatography. So here in chromatography we have a filter paper or the chromatography paper, and on this filter paper we put a drop of blue ink, and then the filter paper is kept inside a liquid. Now care has to be taken that the liquid, this liquid does not touch. See, the level of the water is only till here. It does not touch the drop of ink. So here is my ink, and as time passes, just to make it easier, I just have made it into blue ink. I've made it the black ink, and here this is water. Now as time passes, water begins to slowly rise into this filter paper by the capillary action, and it slowly keeps rising up. And as the water rises into this filter paper, to your surprise, you will find now the ink is now split into this little many spots. There's a green here, there is also a blue, there is red, and there is yellow. So it gets split into many different colors. So now we can conclude that this black ink was made up of all these components. Now why did this component separate out? Because as the water began to rise into the filter paper, it dissolved the black ink. Here in this case it dissolved, and it kept rising. This green color had the least solubility, and because it had the least solubility, it did not rise very quickly with the water, so it remained at the bottom. Then came the purple. The purple had a little more solubility than the green. So all these different components, they dissolved in water, but the solubilities were different. The ones with least solubility remained at the bottom, while those with more solubility rose very quickly with the water. So in this diagram we see that the yellow color, which is right at the end, so this yellow will have maximum solubility because it rises very quickly with water. So we can define chromatography is a technique used for separation of solutes that dissolve in the same solvent. But I think we could add something. The solutes have different solubilities. And because the solutes have different solubilities, and because of this difference in solubilities, they get separated out into different components. We'll now study the principle of chromatography. Why does chromatography take place? The main reason is because of the difference in solubility. So this is what I've taken from your textbook. This is the principle of chromatography, exactly taken as of picture of your textbook. It is the colored component that is more soluble in water rises faster. In this way, the colors get separated. Here are some applications of chromatography to separate colors in a dye. Like here, can you see? Here, these are different dyes, and by carrying on the chromatography, we can see the different components of that dye. Pigments from natural colors, drugs from blood. Here also another good example of chromatography of plants. Here we can make out the different colors which are obtained from the chlorophyll. We can know the different components present in the plant. It can also be used to separate drugs from blood. If the chromatography of blood is carried out, and if a person has consumed drugs, then that paper, that filter paper, the chromatograph paper, shows a clear strip of the components. So you'll have like different strips here. It won't be this color. This is just to explain to you. You'll have different strips, and then the laboratory will analyze these different strips. They will study the different strip, because each strip, each color means there is one component, and on studying the strip they will know if there is drugs or poisons or other unusual substances present in the blood.